Hi, I'm Scott Bartell from the University of California, Irvine Program in Public Health. This is joint work with collaborators at Drexel University and Emory University. So let me start with a little bit of the motivation and importance of this work. We are very interested in risk assessment and development of regulatory standards using animal and human data. In some cases, for many toxicants, in fact, both types of data sources are available for doing dose-response analysis and risk assessment. But quite often, regulatory agencies pick one type of data or the other rather than trying to combine them to do quantitative dose-response analysis. Of course, animal studies use randomized assignment to treatment normally, so there are few threats to validity internally, but questionable relevance for actually estimating dose-response relationships in humans from those data. Human studies, of course, are conducted in more relevant populations, but they're typically conducted using observational designs. And as we know, exposure and confounders can be quite difficult to identify and measure accurately in these study designs. So quantitative methods are really needed for utilizing this dose-response information from both animal and human studies in a way that actually respects the relative strengths and weaknesses of those two types of data sources. So in this paper, we actually use data on silica and lung cancer from previously published sources to demonstrate how one might go about combining these in a way that makes sense. The human data come from nested case control sample from 10 pooled cohorts representing nearly 66,000 workers and over 1,000 lung cancer deaths. And we sampled a nested case control sample from these to make it more efficient, matching on cohort, sex, race, and age within 5 years. Animal data were obtained from 3 randomized trials. We pooled nearly 600 rats and 49 lung cancers from those studies in order to characterize the animal data on silica and lung cancer. So the central theme of this paper is using Bayesian methods to combine these two data sources. So this begins with prior specification, represented by the equation shown on this slide, which is just the idea that the hazard ratio in the human is equal to the hazard ratio in the rat times some uncertainty factor, where in the Bayesian framework you use a probability model to represent that uncertainty, reflecting either 1, 3, or 10-fold uncertainty for cross-species extrapolation. These ideas of actually the 3-fold and 10-fold have been around for quite some time in EPA risk assessments and other toxicological risk assessment. And a recent National Research Council report on risk assessment at EPA suggested converting those to this Bayesian framework. We also use in our paper a measurement error model to account for Bergson measurement error, which we expect would affect the validity of the human studies due to the use of group-based job exposure matrices assignments for the individual exposure ascertainment. We then used R with JAGs to update the prior with the human data and to obtain posterior estimates for the hazard ratio in humans. There's very detailed results in the paper and several tables, but just to briefly summarize here, our posterior means shifted towards the rat hazard ratio when we assigned less uncertainty for the animal to human extrapolation, or when we use single human studies instead of the pooled data set from all 10 studies. And this is kind of consistent with what we'd expect from Bayesian theory, which is that with smaller sample sizes on the data, we put more weight on the prior. We also noted that accounting for Bergson measurement error decreased the weight given to the human data, affecting the hazard coefficients for some analyses, but not appreciably affecting them for others. Notably, the posteriors were more resilient to measurement error for the log-transformed exposures than for untransformed exposures. There's a lot more great detail in the paper, so I encourage you to read it, and please don't hesitate to contact me if you have any questions.